Okay, can everybody see my slides? And yeah, okay,
Yes. Yes.
great. And feel free to stop me anywhere in between to ask questions. And uh, and you could also save it to the end, but feel free to stop me in between. Okay. So Sonia talked to you in the last two classes about the diversity of the neurons and how the entire complex nervous system has evolved sort of from very simple systems like Hydra, where is Hydra to Drosophila to C. elegans to mammals like mouse, and then finally the humans. And how it is all assembled into circuitry, and how this is important to produce behavior and perhaps a little inkling of how did it evolve. So I will now, and she's already told you all this from the invertebrate nervous system. So today I will take you through the vertebrate nervous system, and my talk is also structured very similar to hers, in the sense that she began by telling you how you specify the neurogenic region in the developing embryo, and then how you then go on to do a very important thing in the developing embryo, that is patterning, spatial patterning, which is the first thing which happens, which is happening across at least two major axes in the developing embryo. One of them being the dorsoventral, and if you can see me, the dorsoventral is this, where this is the top of the head and this is the bottom, and the anterior posterior is simply just the face and the back of the head. Um, and then she then told you some, and I think one of the very important, and once you have the areas or the different areas in the brain generated, what you really need locally is divisions of the stem cells. So one hallmark of the entire nervous system development is the fact that all the diverse neurons which you see, be it the mouse or the humans, the billion neurons you see in the humans, they all come from a common pool of stem cells, right? Which themselves undergo restriction in terms of different kinds of progenitors, which I'll be talking about in my next class. She walked you then, once she talked about the area patterning, she also told you about the division, and particularly the very important asymmetric division, which is the division of the stem cell into generating one more stem cell and a differentiated progeny, which then goes on to become a neuron or a glia in the nervous system. And a key aspect of this division and generation of the neurons in glia is the temporal patterning. So I'll be covering those parts in the second class, which we have on Monday. But today I'll be talking to you about how is the entire nervous system, and at least the first initial areas across the axes, generated. Okay. Now, before we begin, I thought I will show you this, which comes from I call him sort of the, quintessential developmental biologist, which is Scott Gilbert. And I'm sure all of you would have seen read his book, particularly in this, during this course. And he, in one of his articles, where somebody asked him that, what would you like to sum up as a biologist? And he said that all I really need to know from biology, I learned it during gastrulation. And what is true for gastrulation is perhaps true for everything in development. And a few things which I think one of the key points which I took from his article here, and which I'll be sharing with you after my talk. And I suggest all of you read it before your course ends, because all that which you've read till now, probably he has summarized it very well in this article. But one of the key things which was a takeaway for me was the fact that he said there are three rules, fundamental rules of development. And he said the first is timing is everything. And that perhaps alludes to the temporal timing or regulation of this entire process. The next thing he said that there are three determinants of value. And he said it's location, 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 which means location is the key. That is the spatial angle to this. And lastly, perhaps it's a joke, he just says that both of the above mentioned statements are true, which means that two things which are very important in development is timing and location. And that's what determines, I think, everything else. That start of the timing and the location determines everything else, everything in development, and very, very true for the nervous system development as well. Right. I'm going to show you here two videos just to sort of give you a sense of how the embryo develops. And these are from the HHMI websites where they have an online biointeractive. And I suggest that you go and visit to look at some of other videos which they have. I'm not sure. I'm going to play this, and I don't know whether you'll be able to hear the audio. Just let me know. Otherwise, I'll give a voiceover to this. So here, what is going to happen is just they're going to show you how the embryo development happens. And I thought it's good to see it in a 3D view so that you have an idea of, when I talk of axes or when I talk of the embryo, you have an understanding of what is the 3D view of the entire process.
In this video, then we'll look at the beginning of development.
Sorry, let me just pause. Are you able to hear the audio from this video?
Yes, but it's very light, it's feeble.
Okay. Let's try again.
Looking into the ovary at an
How's it
Now
now?
it's okay.
No, it's okay. Okay. I'll just start it again.
Then we'll look at the beginning of development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus, and then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days. At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc. And then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the three beginning layers of the embryo, the so-called germ layers, are formed. And I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns and appendages start to bud out. You see the head forming and the eye. And then eventually we get a small embryo. And some months later, of course, this would be born as a young baby.
Okay, so what I wanted you to maybe sort of bear in mind, because some of my slides going forward is going to perhaps start from this very key step, which during gastrulation is the formation of the three germ layers, and then from there how the specific neural ectoderm comes about. So I just wanted to you all to have a sense of the 3D view of how this entire thing develops. And I'm also going to show you.
Then we'll look at the beginning of.
Sorry, I'm also going to show here is specifically how the human brain development happens, and this sort of will give you. This video will give you a sense of not just the 3D view, but also give a sense of what starts as even smaller than a dime goes on to make this entire gigantic human brain. And all this happens over a period of 9 months. So, I mean, what I really would like you all to appreciate is it all starts from a small pool of stem cells, and that builds the entire embryo and the entire human brain, which we see. Okay, let me start this video.
So we're introducing ourselves into this process about 1 month after fertilization, when the entire embryo is smaller than the size of a dime. And we're going to look in at the nervous system where relatively small numbers of cells have been allocated, and then monitor over the 9 months of human gestation the increase in the number of cells and the change in the shape of the central nervous system. So by 3 months, we have something like 1 million cells. The nervous system is beginning to exceed the size of the dime. You can see that the future forebrain and the cortical region are expanding disproportionately to the rest of the nervous system. By 6 months, we have hundreds of millions of neurons, and the brain is beginning to take now a familiar shape. You can see the beginnings of the indentations, the sulci and gyri that Eric described, so that by the time we approach the 8 and 9 months, this is now a familiar picture, and we have tens of billions of nerve cells present. After birth, there's yet further addition of cells to produce the 100 billion or so nerve cells that occupy the human brain. And one remarkable feature is that even in adult life, new neurons are being added. And there is evidence that the rate of new neuronal production depends on the degree of richness of your environment. So every time you come to the Howard Hughes Medical Institute head.
Okay, so he goes on to talk about adult stem cells.
So
and,
we're,
sorry, okay, right. What are some of the early steps in nervous system development? Perhaps just to, even before we begin, maybe this is a revision of what you learned in the previous classes from Sonia, is the fact that there is the induction and between invertebrates and vertebrates, the induction of the neural plate is perhaps a very common mechanism where the formation and then which in the vertebrates, the formation of the neural tube are perhaps a few, some mechanisms are conserved, and that this process which happens at the very beginning is the induction of the neural plate and neural tube are such mechanisms which are conserved across evolution. Okay, now, right. What I showed you, if you would remember from the video, is that for the formation of the nervous system, I think one of the first, at least the way I understand embryonic development is what perhaps started from the 8-cell stage itself triggers the next steps. So which means the fact that the nervous system development starts from perhaps at the time when gastrulation is happening. And one of the key steps in gastrulation is formation of the midline. So the gastrulation, as you all would have learned in some of your previous classes in DevBio, is the formation of the 3 germ layers, the ectoderm, mesoderm, and the endoderm. And what is quite important at this stage for the formation of the neural plate in the ectoderm is this formation of this region, this zone here, which is called the notochord, which is formed, as you can see here, it is formed by the invagination of the mesoderm. So what you see here is a dorsal view, like you looking at it from the top. And then if you cut the section, so that will be a cross-sectional view, which you can see here, right? Okay, in all my slides which are going to come, all this early image patterning events are all taken from 3 textbooks, Principles of Neural Science, Dale Purves, Neuroscience and Square, Fundamental Neuroscience. And otherwise, if it's not mentioned, it is from those, the sources from those. And I've also taken some papers to highlight some of the important molecules which function in the patterning. Right. So as I was saying, that the first important step is perhaps starts from as the gastrulation ends and as the neurulation begins. So this entire process is called neurulation. So that begins with the formation of the notochord, which comes from the invagination of the mesoderm. And this notochord then forms the midline of the gastrulating embryo. And then just where the midline is, as you can see here, forms the, this notochord induces then in the ectoderm, the formation of the neural plate in the midline and formation of the primitive streak as well. So once this neural plate is induced, then this neural plate. So it is like this. If you can see my video, it is this neural plate. So there is this notochord beneath, and this sends signals into these developing ectoderm, and the neural plate is formed here. And this neural plate then folds on itself like this to form this tube, which then extends from the entire anterior to the posterior. And as you can see here, this is day 20. You would see that in the cross-sectional view, the notochord has induced a floor plate here, and this ectoderm is then coming together to form a tube. And once this ectoderm comes together, the very lateral edges of this ectoderm form a set of cells called the neural crest cells. Now, what are the neural crest cells? So this nervous system in the entire vertebrate consists of two aspects. It consists of the central nervous system, which is the brain and the spinal cord. So anything which comes out from the neural tube is the central nervous system, which at the very anterior, that's towards the face end, forms your entire cortex, cerebral cortex, which does the higher order functions. And then it goes on to form, which I'll be telling you, the other regions in the brain, followed by the spinal cord towards the very posterior end. Now, the neural crest cell are the origin or the source for all your peripheral nervous system. Now, what is the peripheral nervous system? So the peripheral nervous system is all those set of nerves which connect the brain and the spinal cord to the rest of the body. And these peripheral nervous system comes from these neural crest cells. So the neural tube is formed here, and the neural crest cells are present here. And once this is formed, you have, the somites are formed from the mesodermal lineages, and the somites are the ones which give rise to the skeletal and the musculoskeletal lineage, which supports the spinal cord, right? So as you can see here, so in the dorsoventral axis, it is the neural tube, the neural crest on top. There is the floor plate, and there is also a roof plate, which also signals, and I'll come to that in a bit. But also what I want you to understand that there is also another axis of development, which is the anterior and the posterior. So the anterior here, and the folding actually starts from the middle. So what folds first is the middle of the tube, and then in a zipping action, the entire tube is folded like this. So as you can see here, the anterior neural fold is still closing, so is the posterior, and this region here will go on to form the spinal cord, and that region here will go on to form the forebrain of the nervous system, right? So this is just a simplified view, but I wanted to show a TEM of this to show the notochord, the closed tube, and you can see the mesoderm and the neural tube here. Okay, just to give you another sense of this is the view from the entire embryo and how it looks like. So if you would see the entire embryo from the side, what you would see is that this ectoderm here goes on to form the top. The red here is your neural cells, the neural tube, and this is the top view. As you can see that as gastrulation finishes and neurulation begins, this is the tube which is going to fold into itself, right? Okay, now a classic experiment which went on to win the Nobel Prize is the experiment by Spearman and Mangold, where they wanted to understand that if you would like to replicate this axis, particularly the axis so what they really wanted to understand is how this entire patterning happens. Is there some signaling center which, if transplanted, can it make another axis or another head, so as to say? So they did this experiment where they transplanted this dorsal end of the tissue and transplanted it towards the ventral end with the idea that can they now produce two heads. And as you can see here, this is a developing frog embryo, and the moment you transplant this dorsal blastopore lip, you get another axis. And when you cut a section of this axis, not only do you see the entire head, but you do see a secondary dorsal axis, which also consists of the same neural tube. It consists of the somite, it consists of the notochord. So what I really want to tell you here is that what patterns the entire embryo is initially, to begin with, is some of these organizer centers. And one classic organizer center is the Spearman organizer, with which you can actually duplicate axes in the developing embryo, and to create dual neural tubes as well. Okay. In the vertebrate nervous system, as I said, the tube extends from the anterior, and here, as you can see, the top is the anterior. This is again a top view now, where you see the anterior end here and the posterior end here. And once the neural tube is formed, it then undergoes several stages. So important thing to bear in mind is that it's a hollow tube. And the question then comes is, why is it a hollow tube and what is it filled with? So this hollow tube consists, we call them the ventricles in the mammalian nervous system, and it is filled with the cerebrospinal fluid, and adjacent to these ventricles are all your stem cells. And the stem cells, the ventricles, the stem cells line up the entire tube. These are stem cells of the forebrain, these are stem cells of the midbrain, these are stem cells of the hindbrain, and these are all stem cells of the spinal cord. And all these stem cells then give rise to the different kind of neurons which you see across the entire axis. So the neurulation proceeds in mammalian systems by a couple of stages. The first stage is the 3-vesicle stage, where you have broad areas which then go on to form the different regions of the brain. So as I was saying, the anterior to posterior axis consists of the forebrain, which consists of the major region in the forebrain is your cerebral cortex, which has the 6-layered neocortex, where all your learning, memory, motor control, everything, all higher order functions happen. This is then followed by the forebrain, followed by the midbrain, where I'll come to that in a bit, that what are the other regions in the brain. So the forebrain, followed by the midbrain, followed by the hindbrain, and followed by the spinal cord. So very broadly, in the 3-vesicle stage, you have the proencephalon, which gives rise to the forebrain, the mesencephalon, which will give rise to the midbrain, the rhomencephalon will give rise to the hindbrain, and the caudal neural tube forms the spinal cord. And very soon you'd see, as development progresses, that once this is set, this anterior posterior axis is set, local events shape the patterning of the regions locally. For instance, there are centers, signaling centers, which are present at the very end here. There are signaling centers which are present at the lateral end, which will then pattern this entire region into the telencephalon, which forms the cortex. And also the midbrain, the boundary between the forebrain and the midbrain also forms the neural retina, which then forms the optic cup and the lens. Then the midbrain is formed just right adjacent behind it, and then followed by the metencephalon, which gives rise to pons and cerebellum, which are regions involved in regulating movement, and these regulate some of the involuntary control of your body. And this is followed by the formation of the myelencephalon, which is form medulla, and the central canal forms the spinal cord. And this is just to show the neural tube, and you can see here the somites, which forms the musculoskeletal system. Okay.
I have a question.
Yes. Yes.
So is the embryonic cerebrospinal fluid secreted by the stem cells which line the cavities?
No. So the CSF is secreted by a developing region, which is the choroid plexus.
Okay, and are those differentiated cells?
Very good question. So to begin with, the choroid plexus is also a neuroepithelium. And I must say that the choroid plexus, so there are several structures in the embryonic brain which are temporary. The choroid plexus itself differentiates. So it starts with being a neuroepithelium, and then goes on to differentiate to make cuboidal cells which secrete the cerebrospinal fluid.
So can I call it partially differentiated?
I'm not sure how. I mean, I'm really not aware of what. Uh, so I would say that, the neuroepithelium, when it's the choroid plexus neuroepithelium, it is still stem cell.
Okay.
I'm not aware of the divisions or how it differentiates into the cuboidal cells. Maybe, I mean, yeah, I mean, you can probably look up into how this choroid plexus develops in the embryonic brain.
Yeah, sure.
Yeah.
Okay.
So, yeah, I mean, so I think from the question that you raised, I think it's important to understand that not the stem cells across the entire anterior posterior axis in different regions of the brain all have very different molecular signatures, different properties, different morphologies as well. For instance, I think the progenitors which are seen in the retina are very different from the progenitors seen in the forebrain. But what I really want you to understand is that it all starts from a common neuroepithelium. And then as patterning happens, which I'll come to in a bit, that as patterning happens, as regionally the signaling centers send signals, the entire nature of this entire development keeps changing from the anterior posterior axis or the dorsoventral axis. Yeah. Okay. So as I said, the 3 vesicle stage then goes on to the 5 vesicle stage, and shown here is what are the major derivatives from some of these vesicles. So the forebrain then goes on to form the telencephalon, which then goes on to make the cerebral cortex. Also, it is important where it is also the region where the hippocampus is formed. It's the region where the amygdala is formed, which is involved in modulating fear. And then the olfactory bulb is formed at the very, very rostral end of this telencephalon. And the olfactory bulb is where all your neurons from the nose, the olfaction, they come, and your sense of smelling happens here in the olfactory bulb. The diencephalon actually consists of important regions. Mainly, it consists of the thalamus, which is involved in integrating the. What really needs to happen is that inputs from the cortex need to go to your entire body, and similarly from all your sensory modalities, be it hearing, vision, or the sense of touch, tongue, olfaction, everything has to come from the outside world. It comes via the thalamus and reaches the cortex, where you can make high-end decisions on this. The diencephalon also has other regions like the hypothalamus, which controls your body temperature settings and all of that. As I said, the regions like pons and cerebellum, they control your involuntary movements. The medulla is also involved in gait movements and all of that. And the spinal cord, of course, it regulates all the movement in the entire body. Are these questions or something which I'm getting? Oh, okay, that was Sonia. Okay. Right. Okay. And now that we have had a brief overview of how this entire region patterns, what I would like to show from here is, so what I've shown you till now is how this entire stretch tube forms and everything. But what patterns this tube? I think I would say that it's a mix of both signals, but initially to begin with, it's signaling centers which pattern the regions. And once the regions begin to pattern, there are cell intrinsic mechanisms, particularly the expression of transcription factors, which then take on this forward. And that's how you end up having the different patterning in the brain. And so I've shown here is such signaling centers. One such is the notochord itself, because the very beginning, it's only the notochord. And the notochord sends signals to say that, anything on top of me has to now become the neural ectoderm. So the notochord is one such signaling center, and I'll come to in a bit that what the notochord secretes. This is then followed by the formation of the floor plate, which is another signaling center, particularly very important in the in the development of the spinal cord. The mesoderm also actually secretes a lot of signals, and I'll not be talking about them, but the mesoderm is also known to secrete signals which patterns this entire area. And then, as I said, the cells itself, the neural tube cells itself, once patterned, they initiate a cascade of gene expressions of transcription factors, of signaling molecules, and so on and so forth, which themselves then begin to pattern. So what starts as a cell non-autonomous soon becomes a cell-intrinsic mechanism. And then there is this crosstalk between the two, which then patterns the entire brain. So I'm now going to give you examples of 4 such important signaling molecules. These are just signaling molecules which are very, very important in patterning the very first few steps of the nervous system development, and then give you examples of how this patterning happens in the entire tube, maybe across the anterior, posterior end, or the dorsoventral end, right? I think it will not be wrong to say that, what's shown here in this slide are D molecules, which does not pattern the nervous system, but perhaps pattern a whole bunch of other organs as well. But particularly in this case, these 5 signaling molecules are very, very important to pattern the nervous system. And those are the sonic hedgehog, which is, and I'll come to in a bit from where it is secreted. So the sonic hedgehog mechanism essentially involves the binding of sonic hedgehog to patched protein, which then relieves the repression from smoothen then goes on to activate both a repressor gli or an activator gli, which then goes on to bind to the DNA and activates or represses genes. Similarly, the retinoic acid binds to a retinoic acid binding protein, which then has a co-factor, CoA, which then binds to DIN again, activates the gene expression. The FGF has a receptor tyrosine kinase. Then the RAS-MAP kinase pathways is activated, which is phosphorylation. And maybe you will study some of these in great detail in your cell biology courses. But I'm just putting it out here just to tell you how this, so essentially I think one common theme is the signal goes inside the cell, the cytoplasm, activates a few downstream things, which then goes to the nucleus and activates DNA. And so same is true with BMP and SMADs and WNTs and LEF and beta-catenin, and everything goes to the DNA to activate. And this is what I meant when I said signals reach the neural tube cells, the neural epithelial cells, which are lining the neural tube, activates DNA, a whole bunch of target genes, and then that begins a cell intrinsic program in these cells. Right. So one such, I talked to you about the BMP. So the very first step in this process, the very first question to ask is then what determines what is the anterior to the posterior end? And the Spearman study has already shown you that the dorsalizing center, or the center which consists of the dorsal DBL or the dorsal blastopore lip, seems to be very important for this, because if you would remove this and transplant it here, you have a second axis, or the neural tube formed, a second axis formed from the anterior side. So which means that there is some signal which is present in this entire anterior-posterior end, which determines what is a dorsal and what is a ventral center. And the signaling in this case is the BMP signaling, which is very high ventrally and low dorsally. And how it is kept low dorsally is the inhibitors like cordon, noggin, follistatin, cerebrus, and ADMP, which inhibit BMP signaling, and which I think perhaps is shown better here. So This is the ectoderm, which has the BMP signaling. And the moment you have the blocked BMP signaling, so here, as you can see here in the ectoderm, the BMP is secreted, but it's blocked with the inhibitors, the caudin and noggin. And then that tissue goes on to form the neural ectoderm. Whereas if you have inhibition, if you have the BMP signaling, then that forms the epidermal ectoderm, and then also forms the ventral regions in the brain. Okay, and that's how the first level of patterning is set. Right. So that was the anterior posterior axis. Now, what is happening in the dorsoventral axis? So as I said, to begin with, in every axis, it's only inductive signals. And in the dorsoventral axis, as the neural tube closes, and this is towards the spinal cord side. This is towards the spinal cord side. You would see that this is the notochord, which forms the floor plate, induces the floor plate here. And this is the roof plate here. And the roof plate is secreting BMPs, whereas in the spinal cord area, the floor plate is secreting sonic hedgehog. And the presence of the opposing gradient, which is seen better here, perhaps Sonia did talk about the Drosophila neuroectoderm itself having similar such patterning, and the expression is very conserved between the Drosophila and the mouse neural tube. So as I was saying, this is the notochord, and this is the roof plate and the floor plate, and the gradient of BMP and winds on the top and the sonic hedgehog from the bottom is responded by all of these. So all of these are different kinds of progenitors in the entire dorsoventral axis of the spinal cord. They all are SOX2 positive, which is one marker, but they express a wide range of different markers as seen here with different colors from the dorsoventral axis. And then they go on to form the different kinds, subclasses of spinal cords in spinal cord neurons as mentioned here, the V3, the motor neurons, V2, V1, and these are the different classes of spinal cord neurons. So the gradient of sonic hedgehog coming from the bottom and the gradient of BMPs is read by these cells, and they respond to this gradient and thereby make different classes of the spinal cord neurons. Right. So this was at the spinal cord level. But so as I said, different mechanisms and different once your signaling has happened, it's then becomes cell intrinsic. And I'm giving you one example of how a cell intrinsic mechanism creates the region from the hindbrain to the spinal cord. And this is the Hox code. And you would have studied this Hox code during Drosophila development, where it forms the abdominal segments in the Drosophila. And similarly, in the hindbrain, there is this Hox code, which is expressed across the different regions of the developing hindbrain. And this expression then determines the different, segments of the hindbrain, which goes on to generate different kinds of neurons. So this is one such cell intrinsic example, which is coming from the Hox cluster, which is conserved, which makes the abdominal segments in the Drosophila, goes on to make the hindbrain in the mammalian nervous system. Right. The other example, and this is a very, very specific example of the winds, there is this boundary between the midbrain and the hindbrain. And I must tell you this, that these boundary regions are rich source of signaling molecules. And in this case, and they're often quite organizer regions. This is one such organizer region between the midbrain and the hindbrain, which is full of winds, which then pattern, both the midbrain and the hindbrain on either side of the organizer region. And then what really happens is in the deletion mutant of Wnt1, you get a deletion of both the mesencephalon and the metencephalon, which goes on to form midbrain and hindbrain. So important. What this shows is what really is important is these organizer regions, which are found formed at the boundaries are very important to pattern on either side of the developing neural tube. Okay, so till now I talked to you about, how the spinal cord, the dorsoventral axis is generated, the hindbrain. Because we only have 2 classes, I've not covered everything, but I'm just giving you some salient examples of each of these so that you get the flavor of how this thing is patterned. So needless to say that it's not just one molecule or one signaling center or one transcription factor which generates all of these areas. But what I'm highlighting to you is some of the key transcription factors or key signaling centers which pattern and which set the pace for the entire region to be patterned. Okay, so I told you about the spinal cord, the hindbrain, and the midbrain hindbrain boundary. What I'm now going to take you into is this entire, the very the front of the brain, which is the forebrain, and how this region is patterned. This is one example where a classic study was done in 2001, and where like the way I told you about the Spearman organizer, the Spearman organizer was such an early organizer that it was able to then create an entire new axis. But there are several such organizers which are more restrictive in nature. Like one example I gave you was this midbrain hindbrain organizer, which is important to create this towards both the side, the midbrain and the hindbrain, and the deletion of which resulted in the deletion of this midbrain and hindbrain. So which means that this is a restrictive organizer, that it's only able to pattern the region around it. So similarly, there exists a signaling center at the very anterior end of the forebrain. So what I'm showing you here in this is an embryonic day 10.5 brain, and this is the very, very rostral end so this is the entire mouse brain at embryonic day 10.5. And this is the region which is marked here with FGF8 in blue. That then, this entire region then goes on to form the cortex. And this here is the nasal placode, which also have FGF8 in them. Nasal placode, meaning the one which will go on to form the nose and the olfactory bulb in the developing brain. So as you can see here, this is called the anterior neural anterior end, which is full of FGF8. So from Elizabeth Grove's lab, Tomomi Shimogori, who was her postdoc back then and now is a celebrated developmental neurobiologist at the Riken Center, she asked the question that what is the function of this FGF8 center, and is it involved in patterning of the forebrain or the cortex specifically? Now, before I go on to say what is patterning of the cortex specifically, what I mean by that, what I mean by the patterning of the cortex is that there is one thing which is conserved between, say, a mouse and the humans, is the fact that in the cortex, you have different regions catering to different sensory modalities. Like you have the frontal lobe, where you have the motor cortex. This is the front of the brain, which is devoted to all your movement and everything. This is then followed by the somatosensory cortex, which is the parietal lobe, which is then devoted to, all your touch and everything. This is then followed by the auditory cortex, and then the visual cortex, which is towards the very back. So this organization is conserved between mouse and humans. And the question she asked is this organization which is this organization, and here if you can see this region here, so this will form the frontal cortex, this will form the parietal, and this will form the occipital cortex. So this is conserved between mouse and humans. And her question was, does this FGF8 signaling, which is happening at embryonic day 10.5, and this is now a postnatal brain where you have the frontal, parietal, and the occipital regions, does this have anything to do with patterning in the anterior posterior end that is making the frontal cortex here, the parietal cortex here, and the occipital cortex here? Turns out she did a fabulous experiment. She's very good at doing these very, electroporation experiments. What she did was she electroporated this towards the back of the brain. And then, so basically this was like a corollary to the Spearman's organizer experiment. So she just electroporated more of this FGF8 towards the back of the brain and asked the question, how is now the patterning across the anterior posterior end changing? So this is a section here where, so this FGF8 is expressed only at 10.5, it decreases by 13.5. But as you can see here in this 13.5 embryo, you don't see much of FGF8, but she then electroporated it towards the back of the brain. Now, Sprouty1 is one of the downstream targets of FGF8. And if you recollect that the FGF signaling itself comes from the outside, goes to the DNA and activates target genes. And one such target gene is Sprouty1. As you can see here, this is the expression of Sprouty1 at the back of the brain. And the moment you overexpress FGF8, you can see an upregulation of this, suggesting that this FGF8 signaling is also functionally active, that's able to activate transcription factors downstream. Now, the interesting and the cool result is all here, where what they've now done is, she has just done an alkaline phosphatase overexpression. This is a control brain now. And I want you to pay attention to two of these molecules, cadherin-6 and cadherin-8. These are cadherin molecules. And their expression, if you would see that, cadherin-6 is low in the frontal cortex and high in the parietal and occipital. Whereas cadherin-8 is high in the frontal and occipital and low in the parietal. But the moment you overexpress FGF8, you suddenly start seeing that this cadherin-8, who's now, you don't even have to look here. What she's done is a very cool experiment, is that she's left one half of the hemisphere absolutely intact and thus done the electroporation only in the other half, just to, you can then just compare across the two hemispheres to see what is happening the moment you overexpress FGF8. So if you overexpress FGF8, the CAD8, whose expression was restricted to the frontal, is now expanded. And this has then pushed the expression of CAD6, which was parietally there, to more towards the occipital. So which means suddenly the anterior posterior axis has shifted if you overexpress FGF8 towards the back of the brain. And this then, they go on to show that indeed some of the regions in postnatal brains are also changed because of this signaling center. So this is one such example of how such signaling centers pattern the brain across the anterior posterior, particularly the forebrain, which is important to make the different regions like the motor cortex, somatosensory cortex, auditory, and the visual cortex in the right places. So, okay, and one more example I would like to leave, to tell you is this in the dorsoventral axis, where FGFR receptors, again, this is the same FGFs is also involved in the dorsoventral patterning. So the dorsoventral means the top of the head and the bottom of the head. So here in this control, what you're seeing here is the top is the dorsal telencephalon, which makes the cortex, and towards the very medial end here is the hippocampus is being formed here, and this entire region goes on to form the neocortex. And all this ventral here, shown here in green, this forms all your ganglionic eminences from where all your interneurons. So the nervous system in mammalian has two kinds of neurons, the excitatory and the inhibitory. The excitatory all come from this dorsal end. Sorry, my cursor is not going. Yeah, from this dorsal end, which is on the top, whereas the inhibitory neurons all come from the bottom, the ventral end. So this patterning is controlled by the FGFR receptors and FGF8. You knock down one, it is redundant, nothing happens. But the moment you knock down one, two, one, what you suddenly see is a loss of ventral structures. The ventral structures are completely gone. And then if you knock down 1, 2, 3, actually you lose the entire telencephalon. All you end up is this here. This here is a signaling center called the HEM, and this is the choroid plexus here, which generates the cerebrospinal fluid. And this is a very medial structure. This is called a dorsal structure, and this is called a ventral structure. So which means that, a combination of these determines not just the dorsoventral identity, but the medial lateral identity as well. So if you knock down, then you end up having only medial. When you knock down all three, you end up having only medial. When you knock down 1 and 2, you lose the ventral identity. And then, so you need all of these to actually make the dorsoventral and the medial lateral axis in the developing brain. Right. Okay, so I'm really come to the end of my today's talk on patterning. So what I've told you is, just to briefly summarize, is how the notochord specifies the the neuroectoderm. This then closes on to form the neural tube. And then you have signaling centers all across the anterior posterior axis. You have the BMPs. You also have the FGFs. In the dorsoventral, you have the sonic hedgehog, and you have the Wnt and BMPs, which then regulate each of these axes to form the different regions in the brain, which is the forebrain, midbrain, hindbrain, and the spinal cord. So what I'm going to now show you is how this then comes about. I mean, what is all of this in terms of evolution, and where are we going to go next in terms of our own class, in the next class, or perhaps in a very rhetorical way, that how is this then mammalian evolution from the mouse to the humans happened, right? So there is this basic plan of the brain, which is, as I told you, that the very rostral or the anterior end consists of the olfactory bulb. It consists of the cerebrum or the forebrain. So these are all terms which are used for the same structures. It's called the cerebrum, it's called the cerebral cortex, which is also called it contains the neocortex and the hippocampus. So that's the cerebrum. It's the telencephalon as well. Then followed by the midbrain, which hindbrain, and the spinal cord. So this is a basic plan. But what I want you to now see in this basic plan, which is perhaps shown well here in a better way, is that as evolution goes, you would see that some of these. So as evolution goes, I think the evolution has gone from the back to the front, and that is evidently seen here. For instance, you would see that some of the earliest organisms have a very developed hindbrain spinal cord structures, and as you go up in evolution, you start seeing that the cerebrum has started taking more space in these brains as it goes into the mammal, right? And one more thing to notice is depending on the usage of a particular system, like for instance, in this case, if the olfaction is very important, as in the case of reptiles, you would see an elongated olfactory bulb, which is seen specifically in the reptiles, right? So, so the point of this is the basic plan is all out there, but what has happened in evolution is different structures have come on top of each other and developed depending on the need of the organism itself. And finally, what has really happened in the mammals, going from mouse to brain, is the fact that going from mouse to a macaque to a human brain, what has really happened is that this region here, which is the cerebral cortex, has expanded immensely, going from the mouse to the human. And what we are going to now look at in the next class is perhaps, I will take you now, now that I've given you the broad plan of how this entire structure comes about in the vertebrates, what I'm now going to talk to you, the way Sonia has also told you about what are this kind of cell divisions which are important in this entire process. As I said, all of these are stem cells, and now once these stem cells are formed, they are patterned and they know their regional identity that they are a stem cell of the forebrain. They then have their intrinsic cues which divide their mode of division, and whether it's symmetric or asymmetric, and decide the kind of neurons they produce. So I'm going to talk to you about the cell division patterns in the developing brain. And I think perhaps I'll focus only in the neocortex, simply because there are a lot of advancements which have happened in very recent years in the understanding of the neocortex division in the stem cells, or the progenitors, which we call them in the neocortex. So I'll be talking to you about the neocortical cell division. And in that, I'll be addressing the question about temporal patterning, and then actually take you to how between mouse and humans itself, this division and the kind of progenitors has changed, and how that has led to the expansion in the humans. So that's going to be our next class. And that's it from me for today. If I'm happy to take questions from all of you. There's the time. Okay. Yeah.
Hello.
Yes.
Am I audible?
Yes. Yes.
So my question is that when you like do electroporation of these molecules and increase their concentration other region
Mhm,
of the brain, and
mhm,
you see a shift in the pattern,
mhm.
but there is also a possibility that that this molecule may not be directly involved in this process, or maybe it only acting like a noise, so it's affecting some other processes because it's a molecule, it has a like a concentration.
Yeah, yeah.
So
So that's a very
do you,
important. Yeah.
yeah,
So can I paraphrase your question, perhaps, right?
ye-
Right.
yes.
So there are two things. I'm not sure if you guys, if you have all learned about there it being necessary and sufficient. Have you all studied this term in developmental context?
No, I'm not aware of it.
Okay. So, I mean, simply put, a necessary and a sufficient molecule is this. If a loss of function leads to a phenotype, then that molecule is necessary. And if a gain of function is gain of function that you overexpress, then which means that is sufficient to induce that particular region. So let me just go back here. Yes. Okay, so what I've not told you here is that this FGF8, if in the mutant, you do lose the entire patterning as well. So that tells you that this FGF8 molecule is a necessary molecule to pattern this this anterior posterior end, right? So answer to your question that whether it's a noise or not, the answer is no, because when you remove FGF8, you don't get the patterning at all, right? Now, why would you want to do a sufficient question? That why do you want to overexpress and see? Because now you want to ask the question is this sufficient enough to induce or pattern the tissue? For instance, if you had, overexpressed FGF8 and nothing happened here. Sorry, my cursor is not moving for some. Yeah, it's now moving. So suppose you overexpress FGF8 at the back of the brain and nothing happened. What is the conclusion which you would do? Right. So there are, for instance, for instance, you know here that if you remove FGF8 from here, the patterning does not happen, and it tells you that FGF8 is necessary to pattern the brain in the anterior posterior axis, right? Now it is not telling you whether you want it. So there are two things. There is one signaling center and there is also a responding tissue, right? So the question you want to
Yes.
ask is whether the signaling is enough to make the responding tissue respond, irrespective of the fact whether the responding tissue is at the anterior end or at the posterior end. And that's what this experiment has answered. So it has told you that no matter what the responding tissue is, the moment you express FGF8, this responding tissue now starts behaving like an anterior tissue. And that's where the sufficiency question becomes very important, your overexpression. And all of this is very, very true in some very early early embryogenesis, early patterning events, because there is this the thing is that very early on, many of these regions are highly plastic, right? For instance, if you would take
Yes.
out the Spearman's organizer and perhaps implant it, say two, or not at the age when the gastrulation is happening, but perhaps several steps down the way, it's quite possible that the tissue may not respond because it's now it's already matured enough, so it's not going to be responding, right? So here in this
Yes.
case, it tells you two things: that this tissue is responsive, and that this tissue, it doesn't matter the identity of the tissue, but the FGF8 expression here will tells you that it's enough to drive the pattern. So the answer to your question is you need to see it in both contexts: how a loss of function study has been done, how a gain of function study has been done. The loss of function tells you whether it's necessary, it's important to induce or pattern, and this sufficient question tells you whether it is enough to induce it. Did that answer your query?
Yeah. Yes.
Okay.
I have a follow-up also. Does the
Yeah.
expression of the receptors for FGF also differ in these regions?
Sorry, say that again.
Uh, does the expression of the FGF8 receptors
Yeah.
also differ in these regions and they somehow influence?
Yeah. So interestingly, the exact receptor for the FGF8 is not known, at least from what I remember that the exact receptor is not known. So
alright.
because there are so many receptors and so many FGFs, it is difficult to make that one-on-one connection between which FGFs receptors respond to which FGF8. So this entire thing is complexed by the fact that there exists many FGFs and many receptors. And that's why and there is also this redundancy, which, as you can see here, that if you only knock down one, nothing happens. You need to knock down both 1 and 2 to actually do the dorsoventral patterning. And in fact, 3 to even disturb that. Right. So this exact one-on-one is still not known, because I think perhaps you could design an in vitro study to do that. But I mean, to do it in the entire mouse brain, it's still a little difficult to know exactly, because these are molecules which diffuse out and they're not restricted within a cell. So you'll not be able to image it just as it happens. And only very recently people are able to I think there's a very recent science paper where they're able to monitor the diffusion of Wnt molecules. I think such techniques will be useful to know the exact receptor to FGF8. Yeah. Hello. Sorry, am I audible?
Yes,
Yeah,
yes.
we can hear
Yes,
you.
yes. Okay, okay. Any other questions? Okay, so if not, then I think I will call it a day, and then I'll send you my slides and some PDFs for additional reading.
I have just one question.
Yes.
You said that the human cerebral cortex is expanded immensely with respect to the mouse. So are the other structures, the midbrain and the hindbrain, fairly similar between the two?
I don't know so I would put it this way. There has been an expansion across the entire axis, but the expansion which is seen from the cerebral cortex is far, far greater than the other regions. So I don't know the exact change. I think there'll be some review addressing that, but very clearly, I think one of the major differences which has come about, not just between mouse and human, but even between, primates and humans, is the fact that the neocortex has expanded much, much more than the other regions.
Okay, got it.
Okay, so then are there any more questions? Okay, if not, should we call it a day? Is Varsha there?
Yes, Bhavna. Yes.
Varsha,
Yeah,
hi.
we can end the class.
Yeah, we can end the class, and then I'll send you the slides and the PDFs, and we'll meet
Yes.
again on Monday then.
Yeah, sure.
Yeah.
See you.
Okay. Thank you, everybody.
Yeah.
Very nice meeting you all. Yeah.
Thank you.
Thank you.
Bye.
Thank you.
Thank you.
Bye.
Thank
Bye.
you.
Bye.
Thank you.
Thanks. Bye.
Bhavna.
Sorry. Hello.